is Allison Delker, and I'm a senior in animal science. For this discussion, I will be discussing the luteinizing hormone in the mare. This paper will discuss the luteinizing hormone and its effects on the mare. What is the luteinizing hormone, how it works, and what it does? Lastly, the discussion of its involvement in the mare will be noted. What is luteinizing hormone? Luteinizing hormone is a hormone released from the pituitary gland. This hormone triggers ovulation and the development of the corpus luteum. Luteinizing hormone also increases the amount of estrogen the follicle cell produces. Luteinizing hormone also maintains the corpus luteum. How does LH work? The estrous cycle is controlled by hormones. According to the University of Texas at Arlington, the ovarian cycle consists of two phases, the follicular phase and the luteal phase. The follicular phase consists of the hormones follicle-stimulating hormone and estrogen. The luteal phase consists of the hormones luteinizing hormone and progesterone. In order for LH to be released, the follicular phase must occur. But for this presentation, we will skip to the luteal phase. The surge of LH causes the follicle on the ovary to rupture or ovulate. Inside the follicle is the ovum, and when the follicle ruptures, a fluid is released from the follicle and carries the ovum with it, which is a chance for help to be fertilized by sperm. Once the fluid leaves the body, of what's left of the follicle becomes engorged with blood, creating the corpus hemorrhagicum. The corpus hemorrhagicum luteinizes to form the corpus luteum in the cycle. The corpus luteum produces progesterone, which inhibits the release of LH in the cycle. Therefore, progesterone's goal is to maintain pregnancy. If the mare does not conceive, prostaglandin is released from the endometrium and causes the corpus luteum to regress. Progesterone levels would then reduce and inhibition of LH on the cycle is reduced as well, and the ovarian cycle would begin again. If the mare conceives, the developing embryo will inhibit the release of prostaglandin, which it also continues to upkeep the corpus luteum. A little fun fact about LH in the mare is that the secretion is longer than most animals. This graph here is just showing what I discussed with the ovarian cycle. So for example, on the left-hand side you have the follicular phase, which is the first part of the ovarian cycle. Step 1, it shows that the primordial follicle changes to the primary follicle. In step 2, it shows the primary follicle changes to the secondary follicle. And lastly, step 3, the secondary follicle converts to the tertiary follicle. Underneath that, you see that FSH and estrogens are the main hormones used in this phase. If you look to the right, you see the luteal phase, which is what we're focusing on. Here it says step 4 is where the tertiary follicle goes into ovulation. For step 5, it's ovulation to the corpus luteum. Step 6, the corpus luteum changes to the corpus albicans. Again, below this you can see that LH and progesterone are the main hormones in this phase. There are many factors that could go into affecting the levels of LH. Traveling intravaginal sponges and secretions of LH in an intact ovarectomized mare will be discussed in this section. In a study performed by Bacchus and others, mares were artificially inseminated and grouped between a control group and a transport group. The study showed there was no difference between the group with relation to the expression of estrus or hormone levels. Therefore, being worried about conception during the traveling process should not be too high on the list. A study done in 1983 showed that placing an intravaginal polyurethane sponge in the vaginas of anestrus mares increased the concentrations of LH and FSH. A year later, the study was reviewed by Thompson and others. The study viewed 50 horses and looked at hormone levels during the time sponges were placed in the vagina. They concluded that the surges were not always greater than mares without intravaginal sponges. A study performed by Garza and others looked at the secretion of LH in an overactimized mare. Concentrations of LH and FSH were observed in a sample as controlled and a steroid injected. LH seemed to have increased with mares treated with steroids. Therefore, overactimized mares injected with steroids had an increase of LH levels compared to those still intact or not injected with steroids. Overall, this presentation talked about what LH is, which is a hormone produced by the pituitary gland that triggers ovulation and the development of the corpus luteum. We also discussed the functions of LH, which is where the release of LH is part of the luteal phase in the estrous cycle. And we also discussed the factors that are thought to affect the LH levels in the mares. Overall, the luteinizing hormone released in mares is going to have the same levels for the most part in all mares and definitely the same function.